Hi viewers, this is Shomallo Chatterjee. I am currently pursuing my Masters in Biotechnology from Pondicherry Central University. Today, we are going to learn about glycolysis. In last lecture, we discussed about cell, and we know that anything small or anything less than a cell cannot exist independently. We also know that the different life processes which is required for a living organism to survive, all those life processes occurs in the cytoplasm of the cell. But to carry out these life processes on a daily basis, we need energy. Now I'm talking in a micro level. I am talking at a micro level. If I look things from a broad perspective, just think of a human being. We get up every day, we brush, we do the necessary things, then we get ready for the school or college or for office, then we travel there, we do the work, we come back, and then we uh, do a lot of things before going to bed. So, this we do on a regular basis. For this to happen, we need energy, and that energy it comes from food. The food contains different type of biomolecule. For example, it contains carbohydrate, it contains fat, it contains proteins, nucleic acids, vitamins, minerals, a lot of other things, right? But we know that the maximum energy we get from carbohydrate. And we have also learned in the chapter called as digestion that whatever we eat, they are complex food that needs to be broken down into simple absorbable form. And that simple absorbable form in case of carbohydrate, we know it is monohydrate, or we call it as a monosaccharide. And glucose is the simplest name that we can take as an example. So the cell needs energy at every step to survive, to grow, to function, and the only fuel which is available for the cell is glucose. But glucose, as we know, it is a big molecule, it's a six carbon compound that needs to be broken down to extract energy from it. And this is what we call as glycolysis. Let us focus on the name. The name, it contains everything. For example, glycolysis. What is glyco? Glyco indicates sugar. It is not necessarily that it has to be glucose, but glyco indicates sugar. And we know lysis means to break down. So glycolysis is the breaking down of glucose. Normally, what we do is we interchangeably use the term glucose and sugar, but it can be anything. They are basically talking about the monomer, they are talking about the monosaccharide. So, to define glycolysis, it is a metabolic pathway in which one molecule of glucose, which is a six carbon compound, is broken down into two molecules of pyruvate, and pyruvate is a three carbon compound. Okay, now coming to the introduction or coming to the context part. What is glycolysis? We know that it is a metabolic pathway in which one molecule of glucose, which is a six carbon compound, is broken down into two molecules of pyruvate, which is three carbon each. Where the glycolysis occurs, it occurs in the cytoplasm. Very, very important. You should uh, remember the locations. The locations are very important. Glycolysis, it occurs in the cytoplasm. Remember, it do not require oxygen. Oxygen is not a prerequisite, or it is not mandatory. Glycolysis, it can occur in the aerobic condition as well as in the anaerobic condition. So it happens in aerobic as well as anaerobic condition. Now let us look into some of the context as mentioned in the slide. It is also called as Emden-Meyerhof-Parnas pathway. Parnas word is not written. It is also called as a Emden-Meyerhof-Parnas pathway, EMP pathway. Okay. It is universal and most conserved pathway. What do you mean by universal pathway? It occurs in all the living cell. It is something which occurs in all the living cell. That means it occurs in plants, it occurs in animals, it occurs in microbes as well. So it is universal pathway and it is most conserved pathway. Okay. It occurs in the cytosol, as I mentioned, of all the cells of the body, both in aerobic as well as anaerobic condition. It is an emergency energy yielding pathway of the cell in absence of oxygen. So again, the same thing is being repeated, that it occurs in presence of oxygen as well as it occurs in absence of oxygen. It is a major pathway for ATP synthesis in tissues lacking mitochondria, example erythrocytes, that is RBC, red blood cell, cornea, lens, etc. It is essential for the brain, which requires glucose for energy. Okay, so that is the context. Now, before moving on to the pathway, let me give an overview. Okay, I will discuss the pathway in details, but before that, let me give you an overview. After the glycolysis pathway is done, how many steps are there in glycolysis? 10 different steps are there. And each and every step it is controlled by enzyme. Okay, each and every step is controlled by enzyme. At the end of glycolysis pathway, we get ATP, that is adenosine triphosphate. We get NADH and we get pyruvate. Pyruvate, it will further enter into the TCA cycle. Okay, it will further enter into the TCA cycle of the citric acid cycle. Okay, that's why glycolysis is also called as a universal pathway. So this is the first step of cellular respiration. This is the first step of cellular respiration. Again, let me give an overview. We start with glucose, which is a 6-carbon compound. It is broken down into 3-carbon molecule, 3-carbon molecule, that is pyruvate. And the final product that we get is 2-pyruvate. We get ATP and we also get NADH. ATP is being utilized at the same time ATP is produced. So ATP is utilized, it is produced, NADH is produced, and the final product or the major product that we get is pyruvate. 2 molecules of pyruvate we get. So ATP is used, ATP is produced, NAD it is reduced to NADH. Okay. Now let us come to the glycolysis pathway. Again, before going into details, remember that there are 10 steps. All the steps are controlled by enzyme. One half, see, this is divided into 2 different halves. Okay. This 10 steps glycolysis process, it is divided into 2 different halves. One half, the first half, it is called as the energy investment phase. The first half is called as the energy investment phase. That means ATP is used over here. Glucose is being prepared for the breakdown. What is the ultimate goal of glycolysis? To break down the glucose. Now, let me give you a real life example. When you are making a cinema, okay, when you are making a film, before the shooting, we are very much interested in shooting, but there is something called as a pre-production. There is something called as a pre-production. The director, the actors, they go for Reiki. There is a casting happens, then they go for the locations, they choose the locations, they select the locations, and the story is obviously there. Then the dialogue writers come, the script writers come, the costume designers come, then we do the shooting, and finally after the shooting there is something called as a post production. Post production is like the modifications. And let me tell something which is very different from what we are discussing now, uh, because everything is connected. For example, when we go for the protein, okay, the protein undergoes a lot of modification which we call as the post transcriptional modification, post translational modification. So just like that, post production. So here, why I'm saying is, Glycolysis means the breakdown of glucose, but for breaking the glucose, you have to bring certain modifications in the glucose. That modification occurs in the first half of glycolysis, which is called as the energy investment phase. Here, ATP is utilized, and the glucose it is being prepared for breakdown. The glucose we are preparing the glucose. ताम झाम करके इसको प्रिपेयर करना पड़ेगा, फिर इसको जाके तोड़ना पड़ेगा. वो जो प्रिपेयर करने का स्टेप है, that happens in the first half. Let me give another example. When you appear for some examination, you prepare and then you go and give the paper properly, nicely. So that is what is done. The glucose, it is being prepared for the breakdown. Breakdown is the main thing. But before that, preparation should be done. That preparation occurs in the first half, which is the energy investment phase. ATP is used, glucose is prepared for breakdown. Now coming to the energy payoff phase. Energy payoff. From the name only, it is very clear that ATP is produced, NADH is formed, and this is the energy payoff phase. हमने कुछ इन्वेस्ट किया था, अब हमें उसका रिजल्ट, उसका पे ऑफ हमें मिल रहा है. दैट इज द थिंग. ओके, नाउ लेट अस go step by step and we'll understand each and every step properly. Okay, let me take a pen. I hope this will be seen. See, I'm not using the camera because last time after the video I gave in the YouTube, I saw that some things are not visible properly, so that's why I removed the camera. Okay, uh, anyway, let me come to the step by step process. The first one is glucose. We are starting with glucose, which is a 6 carbon compound. See, in this animation, it is very nicely given. See, 1, 2, 3, 4, 5, 6, 6 carbon compound. Okay, so glucose, we are starting with glucose, which is a 6 carbon compound. This glucose, it will be converted to glucose 6 phosphate with the help of an enzyme called as hexokinase. In some books, you will find that instead of hexokinase, something called as a glucokinase is mentioned. Something called as a glucokinase is mentioned. What is the difference between hexokinase and glucokinase? Both of them can convert the glucose into glucose 6 phosphate. But why some books mention glucokinase and some of them mention hexokinase? The difference is not that big. Both of them, they do the same thing. Glucose is converted to glucose 6 phosphate with the help of hexokinase also, with the help of glucokinase also. But glucokinase, it is specifically present in the liver, whereas hexokinase, it is present in most of the tissues. Okay. Now, glucose 6 phosphate, from the name only you can understand at the 6th position, a phosphate group is added. A phosphate is added. So this is glucose 6 phosphate. The first modification is done. This is the first step. The first step is done. The first step, ATP is utilized. Very, very important. In the first step, we are utilizing 1 ATP. ठीक है? पहला स्टेप में हम एक ATP यूज कर रहे हैं. ठीक है? In the first step, we are using 1 ATP. Okay? Now, why we have to add a phosphate? Why the phosphorylation is important? It traps the glucose inside the cell. सेल के अंदर ग्लूकोज को बंद करके रखता है. That's all. It activates the glucose for further reactions. Okay. Now the glucose 6 phosphate will be converted to fructose 6 phosphate with the help of an enzyme which is called as a phosphoglucoisomerase. Phosphoglucoisomerase or phosphoglucose isomerase. ठीक है? ये जो ग्लूकोज 6 फॉस्फेट है, इस ग्लूकोज 6 फॉस्फेट को फ्रक्टोज 6 फॉस्फेट में बदल देगा कौन? आइसोमरेज. Phosphoglucose isomerase. What is happening basically? This step is all about isomerization. So remember the process isomerization. Automatically you will remember the enzyme which is phosphoglucoisomerase. Phosphoglucose isomerase. ठीक है? अब ये वाला स्टेप में what is done in the second step? We started with glucose, we converted it to glucose 6 phosphate, and now the glucose 6 phosphate is converted to fructose 6 phosphate. It is converted from a 6-membered ring to a 5-membered ring. ठीक है? उसको छोटा कर रहे हैं. दिस स्टेप इज डन बाय द एंजाइम फॉस्फोग्लूकोज आइसोमरेज। ठीक है, अब आते हैं थर्ड स्टेप पे। देखो, we started with glucose, we came to glucose 6 phosphate, then this is converted to fructose 6 phosphate, and now the fructose 6 phosphate will be converted to fructose 1,6 bisphosphate. 1,6 bisphosphate. From the name only, it is very clear that सिक्स्थ पोजीशन में एक फॉस्फेट तो ऑलरेडी था, अब पहला पोजीशन में भी एक फॉस्फेट हम अटैच कर देंगे। This is done by the enzyme called as phosphofructokinase or PFK. This is done by something called as a phosphofructokinase or PFK. This step is the most important step in glycolysis. Okay, so if someone asks what is the most important step in glycolysis, this is the third step where the fructose 6 phosphate is converted to fructose 6 phosphate is converted to fructose 1,6 phosphate. That is the most important step in glycolysis. Here another ATP is utilized. तो मैं यहाँ पर एक और ATP लिख देता हूँ। ठीक है, तो पहला स्टेप में जहां पर हम ग्लूकोज को ग्लूकोज 6 फॉस्फेट में कन्वर्ट कर रहे हैं, उधर एक ATP हमें यूज करना पड़ेगा। और ये वाला थर्ड स्टेप में, जो कि सबसे इंपॉर्टेंट स्टेप है ग्लाइकोलिसिस के पूरा जो भी 10 स्टेप्स है, इसमें सबसे इंपॉर्टेंट अगर कोई स्टेप है तो वो थर्ड है, जहां पर हम फ्रुक्टोज 6 फॉस्फेट को फ्रुक्टोज 1,6 बिसफॉस्फेट पे बदल रहे हैं विद द हेल्प ऑफ फॉस्फोफ्रुक्टोकाइनेज। और ये बहुत इंपॉर्टेंट स्टेप है। Why this step is important? Because it is a irreversible step. It is a irreversible step. This is also called as a rate limiting step. This is a irreversible step. This is a rate limiting step. ठीक है, and this is one of the major regulatory step. Why regulatory step? देखो, we all know about enzymes. Okay, अगर हम इस पाथवे को ब्लॉक करना चाहते हैं, सपोज आफ्टर अ रिसर्च वी फाइंड आउट दैट ग्लाइकोलिसिस इज रेस्पॉन्सिबल फॉर अ पर्टिकुलर डिजीज, तो उस केस में हमें ग्लाइकोलिसिस को ब्लॉक करना पड़ेगा। और ग्लाइकोलिसिस को ब्लॉक करने के लिए हम ये वाला स्टेप को यूज करेंगे। दैट इज व्हाई इट इज कॉल्ड एज अ मेजर रेगुलेटरी स्टेप। अगर हम इस एंटायर प्रोसेस को रेगुलेट करना चाहते हैं, अगर हम इस एंटायर प्रोसेस को कंट्रोल करना चाहते हैं, तो फॉस्फोफ्रुक्टोकाइनेज इज द एंजाइम दैट वी आर गोइंग टू टारगेट। द थर्ड स्टेप ऑफ ग्लाइकोलिसिस इज व्हाट वी आर गोइंग टू टारगेट। ओके, now the fructose 1,6 bisphosphate, it breaks down into two different part. Okay, it breaks down into two different part with the help of an enzyme called as aldolase. Okay, phosphofructokinase, it breaks fructose 6 phosphate to fructose 1,6 bisphosphate, और ये fructose 1,6 bisphosphate को हम दो हिस्सों में बांट देंगे। One is called as a DAP, one is called as a G3P with the help of an enzyme called as aldolase. ठीक है, अभी तक 6 कार्बन मॉलिक्यूल, अभी तक वी हैड 6 कार्बन मॉलिक्यूल, राइट। अब इसको हम दो हिस्सों में डिवाइड कर रहे हैं, तीन तीन। ठीक है, अभी तक we were working with 6 carbon molecule, now we have divided it into 3 carbon molecule. ठीक है, one is called as a DAP, one is called as a gap, G3P, or we can also call it as a gap. Now what is the full form of DAP and what is the full form of gap? Okay, let me write it down so that you understand it. See, DAP, DHAP, DAP. DAP stands for what? DAP stands for dihydroxyacetone phosphate. It stands for dihydroxy. I hope it is clear. Dihydroxyacetone phosphate. This stands for dihydroxyacetone phosphate. Okay, it stands for dihydroxyacetone phosphate. It stands for what? It stands for dihydroxyacetone phosphate. And we have something called as a gap, or we also call it as G3P. This is glyceraldehyde, 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 glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate. This is called as a what? Glyceraldehyde 3-phosphate. Okay, So DHAP is quickly converted to G3P, and this conversion it is done by an enzyme called as a triose phosphate isomerase. This is done by an enzyme called as a, ये जो convert हो रहा है ना, ये जो DHAP है, ये जल्दी से convert हो जाएगा G3P में. DHAP will be converted to G3P. यहाँ पर एक एंजाइम होता है, उस एंजाइम का नाम है, इधर नहीं दिया है, मैं बोल दे रहा हूं, ठीक है. इस एंजाइम का नाम क्या है? इस एंजाइम का नाम है, लेट मी चेंज द कलर. इस एंजाइम का नाम है ट्रायोस फॉस्फेट आइसोमरेज. द एंजाइम्स नेम इज ट्रायोस फॉस्फेट आइसोमरेज. ट्रायोस फॉस्फेट आइसोमरेज. व्हाट इज द नेम ऑफ दिस एंजाइम? ट्रायोस फॉस्फेट आइसोमरेज. ये एंजाइम है जो कि डैप को कन्वर्ट करेगा G3P में. ओके, ये एक ऐसा एंजाइम है विच इज गोइंग टू कन्वर्ट, ओके, दिस इज गोइंग टू कन्वर्ट the DHAP into G3P. Okay, triose phosphate isomerase is an enzyme which is going to convert DHAP into G3P. Is that clear? अब ये G3P, now we come to the fifth step. So first step is what? Glucose will be converted to glucose 6 phosphate. Okay, with the help of hexokinase और glucokinase, और इधर हम एक ATP को यूज करेंगे. ठीक है, अब दूसरा स्टेप क्या है? ग्लूकोज 6 फॉस्फेट को हम कन्वर्ट कर रहे हैं फ्रुक्टोज 6 फॉस्फेट में विद द हेल्प ऑफ फॉस्फोग्लूकोज आइसोमरेज. ये हमारा दूसरा स्टेप कंप्लीट. अब तीसरा स्टेप क्या है? फ्रक्टोज 6 फॉस्फेट इज कन्वर्टेड टू फ्रक्टोज 1,6 बेस फॉस्फेट विद द हेल्प ऑफ फॉस्फोफ्रुक्टोकाइनेज, इन शॉर्ट वी कॉल एज PFK. दिस इज द थर्ड स्टेप. और इधर एक और ATP यूज होगा, और ये जो स्टेप है ये बहुत इंपॉर्टेंट स्टेप है, क्योंकि ये स्टेप जो है वो इरिवर्सिबल है. हम फ्रुक्टोज 1,6 बिस फॉस्फेट को बैक करके फ्रुक्टोज 6 फॉस्फेट नहीं बना सकते हैं. तो ये इरिवर्सिबल स्टेप है. दिस इज ऑल्सो द रेट लिमिटिंग स्टेप. और सबसे इंपॉर्टेंट, अगर हमें ग्लाइकोलिसिस को रेगुलेट करना पड़ेगा तो हमें फॉस्फोफ्रुक्टोकाइनेज को टारगेट करना पड़ेगा. ये वाला स्टेप को हमें टारगेट करना पड़ेगा. तो हमें इधर भी एक ATP यूज हो रहा है. दिस इज द थर्ड स्टेप. What is the fourth step? The fructose 1,6 bisphosphate is converted to DHAP and GAP. Okay, DHAP stands for dihydroxyacetone phosphate, GAP stands for glyceraldehyde 3 phosphate, और DHAP जो है वो कन्वर्ट हो जाता है G3P में with the help of an enzyme called as triose phosphate isomerase. ठीक है, अब देखो, अभी तक हमने जो भी देखा है, ठीक है, अभी तक द फर्स्ट स्टेप, सेकंड स्टेप, थर्ड स्टेप एंड फोर्थ स्टेप जो भी देखा है हमने, ये सारे जो है these are part of the energy investment step. All these are actually part of energy investment step. ठीक है, एनर्जी इन्वेस्टमेंट फेज, जहां पर ATP यूज हो रहा है, जहां पर ग्लूकोज को तैयार किया जा रहा है ताकि वो आगे जाके ब्रेक डाउन हो सके. राइट, अब दिस G3P, okay, G3P, it will now be converted. Okay, this G3P will now be converted to, this will be converted to the G3P, it will be converted to 1,3 bisphosphoglycerate. It will be converted to 1,3 bisphosphoglycerate. और इधर जो एंजाइम हमें यूज होगा, जो एंजाइम हमें यूज करना पड़ेगा वो है G3P dehydrogenase. We are not telling the entire thing because it is very big. Glyceraldehyde 3 phosphate dehydrogenase, this is the enzyme which is being used. Okay, G3P is converted to 1,3 bisphosphoglycerate. G3P, it is converted to 1,3 bisphosphoglycerate. इधर बहुत इंपॉर्टेंट स्टेप है ये वाला, क्योंकि यहाँ पर एक NAD प्लस इज कन्वर्टेड टू NADH. ठीक है, दिस इज अ स्टेप वेयर वी गेट NADH. ओके, सो फिफ्थ स्टेप इज वेयर वी गेट अ NADH. ओके, डन. अब 1,3 bisphosphoglycerate will be changed to 3 phosphoglycerate with the help of phosphoglycerate kinase. ओके, और ये वाला स्टेप में 1,3 bisphosphoglycerate, this is the sixth step. Remember, this is the sixth step. 1,3 बिसफॉस्फोग्लिसरेट is changed to 3 फॉस्फोग्लिसरेट. इधर हमें एक एटीपी मिलेगा, इधर हमें एक एटीपी मिलेगा. And one more important thing to remember, this ATP we are getting with the help of a process called as substrate level phosphorylation. The phosphate is transferred directly from ADP to form ATP. Just look very nicely. 1,3 bisphosphoglycerate, it is changed to 3 phosphoglycerate. That means what? 1,3 bisphosphoglycerate, दो फॉस्फेट था इधर, दो फॉस्फेट था, उसमें से एक फॉस्फेट को हम हटा रहे हैं. जो फॉस्फेट पहला कार्बन के साथ अटैच्ड था, वो वाला फॉस्फेट को हम हटा रहे हैं. And that phosphate will join with ADP to form ATP. So this is nothing but substrate level phosphorylation, in short we call as SLP. Since there are two G3P molecule, this step will produce 4 ATP in total. तो ये वाला स्टेप में this step is going to give us 4 ATP. Okay, ये वाला स्टेप में हमें 4 ATP मिलेगा. कितना मिलेगा? 4 ATP मिलेगा. ठीक है. फाइनल प्रोडक्ट देखो, 3 phosphoglycerate now will change to 2 phosphoglycerate. Okay, 3 phosphoglycerate will change to 2 phosphoglycerate with the help of phosphoglycerate mutase. यहाँ पर कुछ नहीं, जस्ट पोजीशन चेंज कर रहा है. ठीक है, और कुछ नहीं हो रहा है. ठीक है. So phosphoglycerate mutase is the enzyme which is going to convert 3 phosphoglycerate to 2 phosphoglycerate. And 2 phosphoglycerate will now be converted to phosphoenol pyruvate with the help of an enzyme called as enolase. With the help of an enzyme called as enolase. Remember, phosphoenol pyruvate, this is the most energy rich molecule. This is the most energy rich molecule. And the final step is the phosphoenol pyruvate gets converted to pyruvate, which is our final product. Okay. And this happens with the help of an enzyme called as pyruvate kinase. Pyruvate kinase is an enzyme which converts the phosphoenol pyruvate to pyruvate. So that is our final step. That is our final product, we can say. The phosphoenol pyruvate, which is the most energy rich molecule, it is getting converted to pyruvate. Okay, it is getting converted to pyruvate. ठीक है. अब देखो, इधर हमें एक एटीपी हम यूज कर रहे हैं, यहां पर हम एक एटीपी यूज कर रहे हैं. ठीक है. फर्स्ट स्टेप में हम एक एटीपी यूज कर रहे हैं. लेट मी चेंज द कलर अगेन. In the first step, we are using 1 ATP. Okay. In the first step, we are using 1 ATP. In the third step, we are using another ATP. So, how many ATP we are using? 2 ATP. 2 ATP we are using. And how many ATP we are getting? How many ATP we are getting? We are getting how many ATP? We are getting 4 ATP. Actually, इधर थोड़ा सा चेंज होगा. मैंने बोला था कि ये वाला स्टेप में हमें 4 एटीपी मिलेगा, इधर एक्चुअली 2 एटीपी मिलेगा. ये वाला स्टेप में हमें 2 एटीपी मिलेगा, जहां पर 1,3 बिसफॉस्फोग्लिसरेट को 3 फॉस्फोग्लिसरेट में ट्रांसफर हो रहा है. ये वाले स्टेप में हमें 2 एटीपी मिलेगा. ठीक है, विद द हेल्प ऑफ सबस्ट्रेट लेवल फॉस्फोराइलेशन. ठीक है. और लास्ट वाला स्टेप में जहां पर फॉस्फोइनॉल पाइरुवेट इज गोइंग टू चेंज टू पाइरुवेट, इधर भी हमें 2 ADP will be converted to 2 ATP. So 2 plus 2, 4 ATP we are getting. Clear? Let me change the color again. So let me change the color again. See, 2 ATP is invested and 4 ATP we get. So what is the net gain of ATP? अगर हम net gain of ATP को हम देखें, what is the net gain of ATP? What is the net gain of ATP? The net gain of ATP is what? 4 minus 2, which is 2. So we are getting 2 ATP. 2 ATP is our net gain. After glycolysis, 2 ATP is our net gain. And how much NADH we are getting in this particular step? जहां पर G3P, that is glyceraldehyde 3-phosphate, is going to convert itself into 1,3 bisphosphoglycerate. In this particular step, we are getting 2 NADH. Here we are getting 2 NADH. And what we get? We get to pyruvate. So these are the products that we get. We get 2 ATP, this is our first product. We get 2 NADH, this is our second product. And we get 2 molecules of pyruvate, which is our third product. Okay, so that is all about glycolysis. I hope it is clear. If you have any problem, let me know in the comment section.